Hello everyone, my name is Woongbi Kwon from the Furst-McNess company. Furst-McNess is an animal nutrition company based off here in the United States, and I'm currently working as an R&D nutritionist, and I am in charge of our company research farm as a research manager. First of all, I would like to thank the host, Chungang University, for giving me this opportunity to share some of the research that I've done for my PhD in University of Illinois. The topic of today's presentation is understanding branched-chain amino acid interactions in growing pigs' diets. Before we started, I want to acknowledge all the research data that I'll be presenting today is from Hans Stein Monogastric Nutrition Lab in University of Illinois. And all the data is part of my PhD dissertation works, and the project was sponsored by Ajinomoto North America. This is the outline of today's presentation. I'll start with a brief introduction about branched-chain amino acid nutrition in pigs, and then I'll talk about the excess dietary leucine effect in growing pigs. And then I'll talk about the effects of valine and isoleucine supplementation to high leucine diets. And then I'll talk about the interaction between tryptophan and leucine. And then I'll talk about the effects of valine, isoleucine, and tryptophan supplementation to corn protein diets. And then I'll give you the take-home messages at the end. What are branched-chain amino acids? Isoleucine, leucine, and valine are categorized as branched-chain amino acid. Of course, these three amino acids are indispensable amino acids for swine. When you look at the chemical structure of these three amino acids, they have similar side chains. Because of this structural similarity, these three amino acids can be metabolized exactly the same way. So let's take a look at more detail about metabolism of these three branched-chain amino acids. In the metabolism of branched-chain amino acid, there are two common enzymes, and all three branched-chain amino acids share these two enzymes in their catabolic pathway. First enzyme is BCAT, branched-chain aminotransferase, and second enzyme is BCKDH, branched-chain alpha-keto acid dehydrogenase complex. Here is another unique feature of branched-chain amino acid. Most of amino acid can be deaminased mainly in the liver. However, for branched-chain amino acid, the first catabolic step with the transamination enzyme cannot occur in the liver. It is mainly in the skeletal muscle. Let's see how branched-chain amino acid can be metabolized in muscle and liver. Like other amino acids, absorbed branched-chain amino acid from small intestine, they are directly going to the liver through a hepatic portal vein. In the liver, these three amino acids are being used for protein synthesis or being transferred to other tissue for protein synthesis. Then the surplus amount of these three amino acids will go to the skeletal muscle and deaminased by transamination enzyme BCAT. As I mentioned, this deamination of branched-chain amino acid in skeletal muscle is unique because all other amino acids will be deaminased in the liver. So this enzyme, BCAT, produces branched-chain alpha-keto acid, KIV, from valine, KMV from isoleucine, and KIC from leucine. And then these BCKA will go back to the liver for the second step of their metabolism by branched chain alpha keto dehydrogenase BCKDH. This step produces three different acyl-CoA from each alpha keto acid. But in the second step of metabolism, KIC, which is the metabolite of leucine, it stimulates this enzyme, BCKDH. That means not only access leucine, but also more and more of isoleucine and valine can be metabolized, even if there are not enough for protein synthesis. Therefore, it is possible that excess leucine can create the antagonism among the branched chain amino acid. Now let's see how much leucine is included in feed ingredients for swine diets. Here is several ingredients that we mainly use in swine diet formulation. If you look at lysine and leucine concentration in these ingredients, especially for corn and corn DDGS, they have almost 4 times more leucine compared to the lysine concentration. Wheat and barley also have higher leucine than lysine concentration, but it is not extreme. Of course, soybean meal contains a lot of leucine, but it also contains a lot of lysine. So if we formulate diets using corn and soybean meal, we don't need to worry about leucine. However, if we use a lot of corn and corn DDGS for our diet formulation, there will be too much leucine in the diet. Previous literatures indicated that if pigs consume excess leucine diets, excess leucine reduced their gross performance, especially average daily feed intake. And they also concluded that the troublemaker in excess leucine diet was KIC. KIC is alpha keto isocarboate, which is the metabolite of leucine. This compound can stimulate the enzymatic degradation of all 3 branched chain amino acids. So they also found that excess leucine reduced availability of isoleucine and valine for protein synthesis as a result of stimulating effects of KIC on branched chain amino acid catabolizing enzyme. Therefore, it is important to know the effects of excess leucine in growing pigs and how to overcome this problem. From here, I want to talk about interaction between branched chain amino acid and tryptophan. And I'll explain why we need to investigate tryptophan effect on branched chain amino acid nutrition. Tryptophan is another indispensable amino acid for swine. It has been considered as an important amino acid because it is involved in feed intake regulation by enhancing serotonin synthesis. However, since tryptophan and all 3 branched chain amino acids are categorized as large neutral amino acid, so they have to share a common uptake transporter LAT1 to across the blood-brain barrier. For this competition, the most important thing for the transporting amino acid by carrier is affinity. And generally, branched chain amino acids have relatively higher affinity than all the other amino acids, including tryptophan. Therefore, it is possible that excess branched chain amino acid, but in this case, excess leucine, may reduce tryptophan uptake into the brain due to the competition for transporters, and this may reduce serotonin synthesis. This possible theory might be the answer of the question why we also need to consider tryptophan effects on brain chain amino acid interactions. Interaction among leucine, tryptophan, and serotonin is well described. When you look at this graph, there was negative correlation between plasma leucine and hypothalamic tryptophan. In addition, there was positive correlation between hypothalamic tryptophan and hypothalamic serotonin from the same study. Now we clearly know that if pig consume excess leucine diet, pig doesn't have enough tryptophan in the brain and cannot synthesize serotonin from hypothalamus. Therefore, it is important to know the interaction among brain chain amino acid and tryptophan for gross performance of pigs, especially for feed intake. So overall objective of this project was to determine effects of excess dietary leucine and effects of dietary isoleucine and valine supplementation, and interaction between dietary tryptophan and leucine, and then to determine effects of dietary isoleucine, valine, and tryptophan supplementation. From here, I want to talk about the first experiment they've conducted to determine effects of excess dietary leucine in growing pigs. The objective of this study was to test the hypothesis that excess leucine affects nitrogen balance and growth performance, reduces serotonin synthesis in the brain, and increases metabolism of all three brain chain amino acids. 40 growing barrels with initial body weight of 30 kilograms were housed in metabolic crates and were assigned to 5 dietary treatments. We formulated base-out diet using less amount of corn and soybean meal and greater amount of wheat and barley because this is the only way we could get the diet containing SID leucine level at the requirement. Otherwise, we will always overformulate diets above the SID leucine requirement. In order to investigate the effects of increasing level of dietary leucine, we use crystalline L-leucine as the source of excess dietary leucine in the diet. Finally, we formulated 5 diets containing 100, 150, 200, 250, or 300% of SID leucine relative to the requirement. To maintain constant food protein level for all diets, we used crystalline glycine. After 7-day adaptation to test diets, total amount of urine and feces were collected for 5 days according to the market-to-market procedure. For sample collection, all pigs were euthanized on day 15 by electrocution. Blood samples were collected on both day 1 and day 15. Tissue samples of brain, liver, and muscle were collected right after euthanization. For gross performance data, initial and final body weight were recorded on day 1 and day 15, and the amount of feed consumption was recorded daily. Here we have results from the first study. This graph indicates the average daily feed intake for 15 days. Blue bar represents SID leucine at the requirement. The orange bar represents 150% SID leucine. The gray bar, 200%, yellow bar, 250%, and sky blue bar, 300% SID leucine relative to the requirement. As I mentioned earlier, excess leucine has negative impact on feed intake in animals. The average daily feed intake linearly decreased as dietary leucine increased. Same pattern was observed on average daily gain data. Linearly decreased average daily gain was mainly due to decreased average daily feed intake. Taken together, gain to feed ratio also linearly decreased as dietary leucine increased. Same trends were observed in nitrogen balance for 5 days. A decreasing trend for nitrogen retention was observed as dietary leucine increased. In addition, biological value of dietary protein linearly decreased as dietary leucine increased. Because of excess leucine, pigs had less nitrogen retention from their diets. Biological value is a good parameter for nitrogen utilization in the body. It is calculated based on absorbed nitrogen by considering urinary output of nitrogen. Unlike gross performance and nitrogen balance data, a linear increase in plasma urea nitrogen was observed as dietary leucine increased. PUN is considered as a rapid parameter of efficiency of amino acid utilization in pigs. Increased PUN means reduced efficiency of amino acid utilization. Even if all pigs were fed isonitrogenic diet, because of excess leucine, the efficiency of amino acid utilization was reduced. Our second hypothesis was excess leucine reduced tryptophan uptake into the brain, and this may reduce serotonin synthesis. So we analyzed serotonin concentration in hypothalamus. As we expected, linear reduction in hypothalamic serotonin was observed as dietary leucine increased. Based on this brain serotonin data, we could also explain why feed intake was linearly decreased as dietary leucine increased. We also analyzed concentrations of BCKA in muscle and liver. As we expected, the KIC, the metabolite of leucine, and the key of stimulating BCKDH enzyme increased linearly as dietary leucine increased in the skeletal muscle. Same pattern was observed in the liver. KIC concentration in the liver linearly increased as dietary leucine increased. Based on these results, increased KIC concentration by excess leucine may increase catabolism of valine and isoleucine, and therefore it creates amino acid imbalance for protein synthesis. In conclusion from this first study, excess dietary leucine reduced gross performance and protein synthesis, and excess dietary leucine reduced hypothalamic serotonin concentration. And excess leucine also affected metabolism of all 3 branched chain amino acids. From here, I want to talk about the second study that we've conducted to investigate the effects of valine and isoleucine supplementation to high leucine diet, because we wanted to find the solution to overcome negative effects of excess leucine. Therefore, in this study, we test the hypothesis that adding more valine and isoleucine will overcome detrimental effects of excess leucine on nitrogen balance and brain chain amino acid metabolism of growing pigs. So we utilized 144 growing barrels with initial body weight of 28.5 kilograms for this time, and they were housed in metabolic crates and were assigned to 18 dietary treatment. We formulated the base-up diet using less amount of corn and soybean meal and greater amount of wheat and barley. It was the similar basal diets that we used in the first experiment. In order to investigate the effects of increasing level of dietary leucine, isoleucine, and valine, we use crystalline L-leucine, L-isoleucine, and L-valine. Finally, we had 2 levels of leucine, 3 levels of isoleucine, and 3 levels of valine. One thing I want to point out that the dietary concentration that I put here with orange color, they are the requirement of each brain chain amino acid from NLC 2012. The 3 levels of each isoleucine and valine represent below the requirement, at the requirement, and above the requirement. Therefore, we had 18 dietary treatments with different SID branched chain amino acid levels. Before we look at the results from second study, since we couldn't find any three-way interactions for all response criteria from this 2x 3x3 vectorial treatment arrangement, I'd like to show you the data based on two-way interaction and main effects. Let's see the nitrogen balance data first. For nitrogen retention, we only found leucine effects. As we added more leucine, nitrogen retention was decreased. For biological value of protein, we had exactly same trend here. As we added more leucine, biological value of protein was decreased, indicating that excess leucine has negative effects on nitrogen balance in growing pigs. How about plasma urea nitrogen? There was similar trend as we saw in the nitrogen balance data, but for this time we also found the valine effect. As we added more leucine, plasma urea nitrogen was increased. However, as we added more valine, plasma urea nitrogen was decreased. Therefore, adding more valine is beneficial in terms of efficiency of amino acid utilization. And this is the data of brain chain amino acid concentrations in skeletal muscle. For all 3 brain chain amino acids, we only found leucine effect. As we added more leucine in the diet, concentrations of all 3 brain chain amino acids in skeletal muscle were decreased. How about brain chain amino acid concentration in the liver? We found only leucine effects here as well. However, unlike skeletal muscle, as we added more leucine, concentrations of all 3 brain chain amino acids in the liver were increased. Based on the data from these 2 different tissues, we can conclude that excess leucine could change the brain chain amino acid composition and their metabolism in the body. Now let's see the plasma-free brain chain amino acid data. For isoleucine, we found the interaction between leucine and isoleucine, leucine effect, and isoleucine effect. As we added more leucine, plasma-free isoleucine decreased. However, as we added more isoleucine, plasma-free isoleucine increased. According to the interaction, the increase is greater if we have low leucine in the diet. How about valine? Exactly the same trend here as well. We found the interaction between leucine and valine, leucine effects and valine effects. Like I mentioned before, these plasma-free isoleucine and valine data indicate that excess leucine has clear negative effects on availability of isoleucine and valine for protein synthesis. Of course, for leucine, it's pretty obvious we found only leucine effects. The more leucine we have in the diet, the more leucine in the plasma. Now, when we look at BCKA in plasma, for KIC, which is the metabolite of leucine, we found leucine effects and valine effect at the same time. It was surprising because we were only expecting increased KIC concentration and excess leucine. But when you look at the right-hand corner, as SID valine to lysine ratio increased, KIC concentration in plasma was clearly decreased. This indicates that adding more valine in the diet could inhibit the catabolizing of branched-chain amino acid. In conclusion, we confirmed the negative effects of excess dietary leucine in growing pigs in terms of nitrogen retention, efficiency of amino acid utilization, and availability of the other two branched-chain amino acids for protein synthesis. However, valine supplementation can partly overcome the negative effects of excess leucine in terms of efficiency of amino acid utilization, and it might reduce the catabolism of branched-chain amino acid by reducing KIC concentration in plasma. From here, I want to talk about the third study that we've conducted to investigate interaction between tryptophan and leucine in growing pigs. The objective of this study was to test the hypothesis that tryptophan supplementation to high leucine diet is needed to prevent drops in serotonin concentration in plasma and hypothalamus, where the serotonin is synthesized, and the tryptophan is needed to maintain growth performance of growing pigs. A total of 144 growing pigs with initial body weight of 28 kilograms was used for this experiment. There were 2 pigs in each pen, and there was a total of 72 pens equipped with the nipple drinker and a feeder. Each treatment had 8 replicate pens. For experimental diets, we formulated similar basal diet that we've already used in experimental 1 and 2. In order to investigate the effects of increasing levels of dietary tryptophan and leucine, we use crystalline L-tryptophan and L-leucine as the source of excess dietary tryptophan and leucine in the diet. We had 3 different levels of tryptophan up to 28% SID tryptophan to lysine ratio, and 3 different levels of leucine all the way up to 300%. Finally, we formulated 9 diets containing 9 different ratios of dietary tryptophan and dietary leucine. Experimental period was 21 days. Blood samples were collected on day 1, 11, and 21. And after the bleeding on day 21, pigs were euthanized by electrocution in order to collect the brain sample. Using blood sample, we analyzed plasma urea nitrogen, plasma amino acid profile, plasma serotonin. Using brain sample, we analyzed serotonin concentration in hypothalamus. For gross performance data, initial and final body weight were recorded on day 0 and 21, and the amount of feed consumption was recorded throughout the experimental period. All the results slide has similar setup. So in the blue, we have 100% SID-leucine to lysine, which is the requirement level of leucine. In orange, we have 200% SID-leucine to lysine ratio. And in gray, we have 300% SID-leucine to lysine ratio. So those are the three different levels of leucine. And here in horizontal axis, we have three different levels of tryptophan from 18, 23, 28% SID tryptophan to lysine. So here you see average daily feed intake data for 21 days of gross performance. As we added more leucine in the diet, there was a pretty dramatic reduction in feed intake. As we added more tryptophan, we saw increase in feed intake, particularly in excess leucine condition. But it was not enough to catch up what we have here with leucine at the requirement, which is the blue bar. Excess leucine for sure reduces feed intake of pigs. For average daily gain, there was no surprise. If pigs don't eat, then they don't grow. So we had exactly same trend here for average daily gain. As we increase leucine above the requirement, we see reduction in average daily gain. As we increase tryptophan above the requirement, we saw some improvement in average daily gain, but it was not enough. We also see here that there is no effect of adding tryptophan if we have leucine exactly the 100% requirement. But in excess leucine condition, there was clear significant effects of adding tryptophan in the diet. What about serotonin? Similar trend as we saw in gross performance data. As we added more leucine in the diet, there was a pretty dramatic reduction in serotonin. However, if we added more tryptophan, we could partly ameliorate that leucine effect, but not completely. From here we have plasma amino acid data. For tryptophan, we saw clear interaction in the plasma. Of course, we added more tryptophan in the diet, we see more tryptophan in the blood. However, there was an interaction, so increase is greater if we have leucine at the requirement in the diet, according to the slope of each line. And for leucine in the plasma, there's no surprise here. The more leucine we have in the diet, the more leucine we have in the plasma. But plasma leucine, in all cases, is reduced by increased tryptophan in diet, indicating that there is interaction between dietary tryptophan and leucine. And when we look at the isoleucine in the plasma, the more leucine we have in the diet, the less isoleucine we have in the plasma. This indicates that excess leucine deaminate some of that isoleucine. Therefore, we get a lower isoleucine concentration in the plasma. That's the same thing for valine. There was an interaction when adding more tryptophan here in isoleucine concentration in plasma. Like I said, same happening here with valine in the plasma. the more leucine we have in the diet, the less valine we have in the plasma because of reduced availability by excess leucine. However, the effects of adding tryptophan was almost zero for valine concentration in the plasma. In conclusion, dietary leucine had negative impacts on average daily feed intake and average daily gain, and dietary leucine reduced hypothalamic serotonin synthesis, and dietary leucine even reduced isoleucine and valine concentration in plasma. On the other hand, dietary tryptophan increased average daily feed intake and average daily gain, and dietary tryptophan increased serotonin concentration. From here, I want to talk about the two separate trials for evaluating the effects of valine, isoleucine, and tryptophan supplementation to high leucine diet. Unlike previous 3 experiments, we used high-protein corn products as the source of excess dietary leucine in these 2 experiments because we wanted to check the effects of excess leucine in more practical condition. So the objective of these experiments were to test the hypothesis that inclusion of dietary valine, isoleucine, and tryptophan will alleviate negative effects of excess leucine from corn protein. For the first trial, we utilized 72 growing barrels with initial body weight of 33.9 kilograms, and they were housed in metabolic crates and were assigned to 8 dietary treatments. For the second part of the study, a total of 288 growing pigs with initial body weight of 28.6 kilograms was used. There were 4 pigs in each pen, and there was a total of 72 pens equipped with the nipple drinker and a feeder. Each treatment had 8 replicate pens. Experimental diet for both trials. We formulated basal diets based on corn and high-protein corn products, which contain 171% SID leucine relative to the requirement. In order to determine the effects of valine, isoleucine, and tryptophan supplementation, we added L-valine only, L-isoleucine only, L-tryptophan only, and combination of valine and isoleucine, combination of valine and tryptophan, combination of isoleucine and tryptophan, and combination of all 3 amino acids. In total, 8 diets were used for the first part of this trial. For the second trial, we formulated one more diet based on corn and soybean meal, which contained 139% SID leucine relative to the requirement as a control diet. Eventually, 9 diets were used in second trial. Like our previous trial, we couldn't find any 3-way interaction from this 2x2x2 vectorial data set. Therefore, I would like to show you the data based on 2-way interaction and main effects. Let's see the nitrogen retention data. We found the interaction between valine and tryptophan and isoleucine only effects. When you look at this graph carefully, adding more tryptophan increased nitrogen retention, and the increase was greater without valine addition, which is the left side from the middle with 70% SID valine to lysine group. However, when we compare numbers with 53% SID isoleucine to lysine versus 63% SID isoleucine to lysine, there were clear reduction of nitrogen retention. This indicates that there is no beneficial effects on adding all 3 amino acids over the requirement. In addition, isoleucine supplementation is even worse. Here we have results from second part of trial and had a different statistical approach. When we look at the average daily gain data, pigs fed the controlled diet had a greater average daily gain compared with pigs fed the other diet. However, it was not statistically different from pigs fed the diet with valine and tryptophan combination. This indicates that combination of extra valine and tryptophan to high leucine diet may be beneficial for negative effects of excess leucine on growth performance. In conclusion from these 2 separate studies, supplementation of valine, isoleucine, and tryptophan altogether was not beneficial, and adding more tryptophan increased nitrogen retention, but the increase was greater without valine addition. But obviously, adding more isoleucine was not great on nitrogen retention. Then adding more valine and tryptophan combination may be beneficial for growth. And this is the take-home message from today's presentation. When you formulate diet for pigs, please don't forget to check leucine concentration in the diet. If there's too much leucine in the diet, especially when you use a lot of corn or corn DDGS for your diet formulation, excess leucine is always bad for the performance. In order to prevent or minimize negative impacts of high leucine, just use more L-tryptophan and L-valine in your diet formulation. However, you need to keep the isoleucine level at the requirement because of the potential negative impact of high leucine and its expensive price. If you have any further questions, please reach out to me through the email, or you can go visit this website, Stein Monogastric Nutrition Lab. You can also get more information about what they are doing for swine nutrition research, and you can subscribe their newsletter on this website as well. Thanks for your attention.